Hello friends, welcome again on Pharma Xn series. In today's tutorial I am discuss about hold time study in pharmaceutical industry. Hold time study perform as per WHO guidelines, supplementary guidelines on GMP, validation, non-sterile process validation. In WHO Expert Committee on Specifications for Pharmaceutical Preparations, 49th Report. Geneva, World Health Organization, 2015, Annex 3, WHO Technical Report Series, number 992. WHO guidelines are intended as a basic guide for use by manufacturers of pharmaceuticals and by GMP inspectors. Before going ahead, let's discuss about introduction of hold time study. Although there are no specific regulations of guidance document on bulk product holding times, good manufacturing practice dictates that holding should be validated to ensure that in-process and bulk product can be held, pending the next processing step, without any adverse effect to the quality of the material. Many times, in industry the material is kept on maximum allowable hold time, which should be established for bulk and in-process drug products. Although regulatory agencies expect manufacturers to document and address hold times, regulatory agencies do not describe a process for establishing hold time practice of sampling, storage for pharmaceutical tablet and injection during manufacturing process. The time period for which the product is on hold shall be justified with adequate data to demonstrate the product will be stable throughout the approved shelf life. In such case the manufacturer shall produce sufficient data demonstrating that the product is stable for said period of time before processing to the next stage, and it will meet the acceptance criteria for the finished product as well as stability specifications. The time during which the product is stored in the bulk container, prior to packing into the final immediate container, constitutes part of the approved shelf life, that is, the date of expiry remains a function of the date of manufacture, not the date of packing. The choice of maximum holding period should be supported by relevant data. Studies may extend beyond the chosen maximum, but it is not necessary to extend testing to determine the extreme limits at which failure occurs. Let's come on definition of hold time study. Hold time study ensure that intermediates and the bulk product can be held without any significant adverse effect on the quality of the material till next step of processing. Such a holding period should be based on data. Studies should not be extended to find the edge of failure for holding. Let's discuss about terminology used in hold time study. First is bulk product. Any pharmaceutical product that has completed all processing stages up to, but not including, final packaging. Second is intermediate. Partly processed product that must undergo further manufacturing steps before it becomes a bulk product. Now I am telling you when hold time study to be performed. New product/exhibit batches. Validation batches. Source of API changes. Changes container and closure system. Manufacturing process changes, etc. Hold time study aspect and procedure to be considered in pharmaceutical industry. Hold time can be considered as the established time period for which materials, dispensed raw materials, intermediates and bulk dosage form awaiting final packaging, may be held under specified conditions and will remain within the defined specifications. Hold time study: establish the time limits of holding the materials at different stages of production by assuring that the quality of the product does not deteriorate during the hold time. To validate the holding time of binders, lubricated granules, tablets, coating suspensions and coated tablets under the prevailing condition, it should be ensured that the result of all process is within the limits of acceptance criteria throughout the holding time. Hold times should normally be determined prior to marketing of a product. The risk assessment of changes in processes, equipment, storage conditions, starting or packaging material should include an assessment of whether further hold time studies should be performed. Hold time study: perform as per approved protocol, as per industrial SOP. No of batch: consider for hold time study to be performed an exhibit or validation as per regulatory recommendation. Typically, one or more batches of a material, intermediate or product can be used for determining hold times. A risk-based approach can be used to determine the appropriate number of batches, considering the characteristics of the materials and other relevant aspects. Samples shall be collected as defined stages and held for defined hold period as per approved protocol. All the samples are stored in simulated container with maintained environmental condition as per defined in approved protocol. After establishing a sampling plan, required sample amount based on the batch size is ascertained, the intervals and tests are to be performed. Samples are kept in a storage container and tested. Results are contrasted and the underlying benchmark information of the control test. Hold time study data shall give the assurance the maximum allowable hold times for bulk and in-process drug products. In general, one lot can be used for validating hold times. If any irregularity results were observed, then another 2 lots can be used for this investigation. Manufacturers may use a flowchart to review the manufacturing procedure for a product and then break up the critical stages of the manufacturing process. Based on the time period required for the particular storage and processing stages, typical pauses in the manufacturing campaign, and the potential impact of storage with reference to environmental and storage conditions. For oral tablets that are coated, the following stages may be considered. Binder preparation to granulation, consider the granulate. Wet granulation to drying, consider the dried granulate. Dried granules to lubrication, blending, consider the lubricated blend. Blend to compression, compression to coating, consider the tablet cores. Coating solution to preparation, consider the coating solution. Coating to packing, consider the bulk coated tablets. Coating to packing in bulk, packing of bulk to finished pack dosage form. Let me show you hold time study flowchart and manufacturing process. Hold time collection of sampling stages are granulation, lubrication, compression, coating suspension, and coated tablets. Look at here, this is a table of sampling stages with time interval when test to be performed. Now I am telling you some important points related to hold time study in pharmaceutical industry. Entire batch shall be hold during process of hold time study. Risk assessment and impact assessment on holding products before hold time study. On the basis of processing time at each individual stage and subsequent stage, we can find the time interval for numbers of tests to be performed. Sample quantity collected for hold time study as per required test in protocol. For the storage of sample, the environmental condition should be same as that of quarantine area/manufacture stage. Where the headspace of containers used for bulk storage in manufacturing and/or quarantine is important, for example, because of a risk of potential degradation as a result of oxidation, then the hold time study should represent worst-case conditions. In such cases, the ratio of headspace to contents in the test container should be at least as great as the maximum that is possible in routine production, especially taking into account part-filled containers. The environmental conditions for sample storage should be the same as those of the quarantine area, manufacture stage. Look at here. This is a list of hold time study protocol, procedure or program should be followed. So we have discussed about the hold time study in pharmaceutical industry in this series. Thank you so much for watching this video.